Hi, and welcome back. This is part 3 of our Chapter 10 series on the biochemistry of the genome. In this video, we'll cover Section 10.3 on RNA and Section 10.4 on the structure and function of cellular genomes. By the end, you should be able to describe RNA structure, the 3 major RNA types, how RNA can act as hereditary material, and also explain genes, chromosome organization, non-coding DNA, plasmids, and viral genomes. RNA is usually single-stranded and shorter than DNA. It's made of ribonucleotides, and these include a ribose sugar instead of deoxyribose sugar, a phosphate group, and a nitrogenous base. RNA uses uracil instead of thymine. And the presence of the hydroxyl group at carbon number 2 in ribose makes RNA less stable than DNA because the hydroxyl group is more reactive. So in DNA, remember, there is no hydroxyl group there. So RNA is more suitable for short-term roles because it is less stable because of this hydroxyl group. And even though it's single-stranded, RNA can fold back onto itself into 3-dimensional shapes that are stabilized by intramolecular base pairing. RNA is usually single-stranded compared to DNA, which is typically double-stranded. We did learn about exceptions in viruses, where some viruses had single-stranded DNA and double-stranded RNA, although this is typically what we see more often in most cells. And for RNA, even though it's single-stranded, it can fold back on itself to form 3-dimensional shapes that are stabilized by so within the same molecule, intramolecular base pairing. So how does the structure of RNA differ from DNA? We know that the sugar is different. RNA contains ribose versus DNA, which contains deoxyribose. And deoxyribose lacks an oxygen at that 2' carbon. For bases, RNA uses uracil and DNA uses thymine. And then in terms of strands, RNA is typically single-stranded, DNA is usually double-stranded. For length and stability, we said RNA molecules are typically shorter and less stable than DNA. And for the shape, RNA can fold into complex 3-dimensional shapes via that intramolecular base pairing. mRNA is a temporary copy of DNA's instructions. It carries gene information to the ribosome, and it's produced through a process known as transcription only when a gene, the DNA portion, is active. In prokaryotes, mRNA is typically short-lived. It doesn't last very long, so this ensures that proteins are only made when they're needed. So you might have seen this before in other books. DNA is transcribed, so I'm just going to abbreviate that transcription to messenger RNA, and then messenger RNA is going to be translated. I'm just going to write the abbreviation, translation to protein or polypeptides, and that's shown here. Right, I have my double-stranded DNA transcribed to my messenger RNA, and then that's going to be translated into protein. Ribosomal RNA, or rRNA, makes up about 60% of the ribosome's mass. So remember, ribosomes are involved in the process of translation, and there was a large subunit and a smaller subunit. So it turns out that ribosomal RNA makes up a large part of that structure. So ribosomal RNA not only provides structural support, but also helps to align messenger RNA and something we'll learn about soon, transfer RNA or tRNA during translation. Most importantly, it catalyzes peptide bond formation between amino acids, making ribosomal RNA a ribozyme. Remember that not all enzymes were proteins. Some of them were rRNA molecules called ribozymes. So that's what we're talking about here. And these ribozymes have both a structural and enzymatic role. Transfer RNA, or tRNA, is also involved in that translation process, reading messenger RNA and producing protein. So tRNA molecules are very small, only around 70 to 90 nucleotides long, but they fold into a characteristic 3-dimensional shape that kind of looks like a T, and I'll show you in a bit. Each tRNA can carry a specific amino acid, and has a region called an anticodon that's going to pair with a codon on messenger RNA. And I'll talk more about that in chapter 11. This will adapt the nucleotide sequence into an amino acid sequence, ensuring that proteins are built correctly. So here in this image of translation, I can see that tRNA is here. We're going to learn later on there's something here called an anticodon that matches a codon in messenger RNA, and there will be an amino acid attached to one end of the tRNA. So here's another look at that tRNA, and it's a pretty cool role that transfer RNA plays. It has this shape that kind of looks like that T. This is a more realistic depiction of what tRNA looks like, but at one end, it's going to be able to bind to an amino acid, and the amino acid is determined by the anticodon present on the other end. So the anticodon, we're going to talk more about in chapter 11, will pair with something known as a codon that's found within the messenger RNA. But transfer RNA has a pretty neat function because it basically helps translate nucleotide information, this is one language, into a different language, amino acid sequences that make up proteins. Our book has a really nice table that compares the structure and function of the different types of RNA. We have messenger RNA, ribosomal RNA, and transfer RNA. For the structure, we saw that messenger RNA was short and unstable because of that extra hydroxyl group at carbon number 2 in the ribose sugar. For RNA, ribosomal RNA, it's longer and more stable, composed of 60% of the ribosome's mass. And for transfer RNA, we saw that its job was to bring amino acids to the growing polypeptide chain, which we're going to talk more about in chapter 11. So for the function, it's going to serve for messenger RNA as an intermediary between DNA and protein. Remember that we only have one copy of our DNA inside the cell when it's not dividing, so it's very precious, right? We want to keep it safe. So whenever we want to make protein, we make an intermediate molecule, the messenger RNA, that can be read to generate proteins. For ribosomal RNA, it plays an important role in translation, and so does tRNA. So ribosomal RNA will align everything together and catalyze that peptide bond formation between amino acids. For transfer RNA, remember it carries the correct amino acid to the growing protein chain. And the table we just saw pretty much summarizes this check your understanding question, the functions of those three major types of RNA molecules involved in protein synthesis. Remember that our messenger RNA, mRNA, was a temporary copy of DNA's instructions, and this carries the genetic information from DNA to ribosomes for protein synthesis. So this was produced through transcription, and it was short-lived. For ribosomal RNA, that was part of the ribosome structure. It aligns messenger RNA and tRNA during translation and catalyzes that peptide bond formation in that growing amino acid chain. Finally, our transfer RNA, these are small RNA molecules that carry amino acids to the ribosome. They're going to carry, we're going to see next time, contains an anticodon region that base pairs with the messenger RNA codon, and it ensures that the correct amino acid sequence is found in the growing polypeptide. We usually think of DNA as hereditary information, but there are exceptions in viruses. So some viruses use an RNA genome instead of DNA. And these can be single-stranded RNA like influenza virus or Ebola virus, or double-stranded RNA like rotavirus. Double-stranded RNA is rare in cells and usually signals viral infection. RNA viruses mutate quickly as well, which really drives their rapid evolution. And here's another table that compares DNA and RNA. This is not from our book, but it's a nice summary of the comparisons of the two molecules. So DNA, remember, was deoxyribonucleic acid, made up of those deoxyribonucleotides. RNA stands for ribonucleic acid, and nucleic acid, that general term, describes both DNA and RNA. The sugar was deoxyribose in DNA, ribose in RNA. For the bases, remember in DNA the difference is we have thymine, whereas in RNA we have uracil. Usually DNA is double-stranded and usually RNA is single-stranded, but not always, like in those viruses. A pairs with T and G pairs with C in DNA. In RNA, there can be pairing. We saw that intramolecular base pairing, but there's no T's, so A would pair with U or uracil in RNA. In DNA, the two strands run anti-parallel, 5 to 3 and 3 to 5, whereas in RNA there's only usually a single strand, although it may fold back on itself. DNA is more stable because of the lack of that OH group, the hydroxyl group, in carbon number 2 of the sugar, and it's also more stable because it's double-stranded. Here it's less stable for RNA because it's single-stranded and because of that hydroxyl group. The roles of DNA are to store hereditary information. We are going to need that when we're replicating DNA as well as when we're producing messenger RNA through transcription. For RNA, we'll see that it has major roles. We saw that in transcription and translation, and we're going to study that further in Chapter 11. We saw that messenger RNA is that intermediate between DNA and protein. For ribosomal RNA, these are catalysts to build or connect amino acids together. And we have tRNA that brings amino acids to the growing chain in the polypeptide. For RNA, it's not usually hereditary information except for some viruses where it can serve as the viral genome. All right, to recap 10.3, we saw that ribonucleotides include a ribose sugar, a phosphate group, and a nitrogenous base. In that ribose sugar, the difference is at the second or 2' carbon, there is a hydroxyl group that we don't see in DNA. There's a phosphate group still on the 5' carbon of the sugar, and for the nitrogenous bases, we have uracil instead of thymine. For the nitrogenous bases, they're still attached to the 1 carbon number 1 on the sugar. And there are still phosphodiester bonds that link our nucleotides together to form RNA strands. For the second bullet point, similarities and differences between RNA and DNA, I kind of covered them, but both of them are polymers of nucleotides. Both of them have that sugar phosphate backbone, and they still have nitrogenous bases, and both can use complementary base pairing for information transfer. We'll see that soon. Differences, we said different sugar. RNA has ribose, DNA has deoxyribose. For bases, RNA uses uracil, DNA uses thymine. Strands, RNA is usually single-stranded, DNA is double-stranded. And stability, RNA is less stable and more short-lived, doesn't have a long lifespan compared to DNA. For the third bullet point, functions of the three main types. So messenger RNA carried genetic information from DNA to ribosomes for protein synthesis. Ribosomal RNA forms the structural and catalytic core of ribosomes, and that catalyzes the peptide bond formation between amino acids. And transfer RNA brings amino acids to the ribosome and matches them to the messenger RNA codon through the anticodon found on the tRNA. And again, we're going to talk more about this in Chapter 11. For the last one, how RNA can serve as hereditary information, certain viruses, RNA viruses, store their genetic material as RNA instead of DNA. And RNA, we saw before, can be replicated directly by RNA-dependent RNA polymerase, and it can also be converted backwards to DNA through that enzyme reverse transcriptase. RNA genomes still carry complete genetic information for building new viruses, and it really demonstrates that DNA is not the only possible molecule of heredity. All right, let's now switch gears and look at our last section for Chapter 10. 10.4 covers the structure and function of cellular genomes. So first, let's cover some relevant vocabulary. A gene is a DNA sequence coding for a protein or an RNA product. A cell's genotype is its full set of genes, whereas its phenotype is the set of traits expressed at a given time. So basically what you see, your physical traits that are seen due to the expression of genes. Some genes are constitutive. These genes are called housekeeping genes sometimes. These are expressed all of the time because they're necessary for basic cell functions. Other genes are regulated and expressed only in response to environmental changes, and those are non-constitutive genes. For example, we'll see in a bit, Serratia marcescens makes a red pigment at cooler temperatures, such as 28 degrees Celsius, or we saw in lab 25 degrees Celsius, but not at 37 degrees Celsius. So Serratia marcescens across these two temperatures have the same genotype, but different phenotype. So in that example I just provided, it tells us that temperature can also regulate gene expression. So our gram-negative bacterium, Serratia marcescens, we've seen in lab produces a red pigment at cooler temperatures, but not at body temperature. Although sometimes in lab we get weird results where the pigment is seen even at warmer temperatures. Oftentimes this is because our incubators are supposed to be at a certain temperature, but they get opened and closed all of the time, so the temperature can fluctuate, leading to abnormal colors that we sometimes see in lab. Let's look at how different organisms organize their chromosomes. In eukaryotes like humans, usually we have linear and multiple and diploid chromosomes. So for instance, you might have seen something like this in other courses. This is called a karyotype, where the chromosomes are lined by size. In humans, we have 23 pairs of chromosomes, and these are 23 from mom and 23 from dad, making that pair or diploid type of chromosomes. In all eukaryotes, DNA is packaged using histone proteins that kind of look like this. So the black thread-like structure represents DNA, and the blue structure represents those histone proteins, so that we can condense and supercoil our DNA enough to fit the nucleus, fit inside that tiny nucleus. So when DNA is wrapped around histone proteins and coiled up, we call that chromatin. It turns out that there's a field known as epigenetics that teaches us how gene expression can change depending on how tightly packed DNA is found, how tightly packed your chromatin is. So if you have very tightly packed chromatin, we call that heterochromatin, like that shown here. These genes are going to be off because enzymes cannot access the DNA. It's kind of hidden within that tightly packed structure. Whereas in euchromatin, the DNA is loosely packed around those histone proteins, allowing enzymes to access the DNA, read those genes, and express those genes. And gene expression means that those DNA genes are going to be actively converted to messenger RNA through transcription and then translated into proteins. In contrast to eukaryotes, prokaryotes have a different type of organization of their chromosomes. They usually just have a single circular chromosome made of double-stranded DNA found in the nucleoid. Remember, prokaryotes do not have a nucleus. These are also usually haploid because they don't have parents, right? They don't have two parents. It's just a cell undergoing binary fission, making an identical copy of itself. So they only have one copy of each gene. They still have to compact their DNA within that tiny cell though, so they're still supercoiling. Although they don't have histone proteins, they have similar proteins. We just call them histone-like proteins. In a later chapter, we're going to talk about enzymes that are involved in supercoiling and the regulation of supercoiling, called DNA gyrase and other types of topoisomerases. And what's also very interesting is that some bacteria, when they infect us, like Helicobacter pylori that causes ulcers, or Shigella, these bacteria can induce epigenetic changes in their hosts, in us, when they infect our cells. And that can change the remodeling of our chromatin, which changes our long-term immune system responses. For this checkpoint, it asks, what is the difference between a cell's genotype and its phenotype? So the genotype is the full set of an organism's genetic information, their entire DNA sequence. The phenotype is what you can see, the observable traits or characteristics resulting from expression of the genotype. And the same genotype can yield different phenotypes under different conditions, like we saw for Serratia marcescens, red at colder temperatures, not red at warmer temperatures. For the second question, how does DNA fit inside cells? So we saw that DNA is supercoiled by enzymes called topoisomerases. We'll talk more about those later. In bacteria, DNA is compacted into that nucleoid region with the help of DNA binding proteins. In eukaryotes, DNA wraps around histone proteins, and we're going to see those structures that look like beads on a string are going to be called nucleosomes in chapter 11. This will further coil to form chromatin, and you see structures called chromosomes right before the cell divides. So this packaging allows DNA molecules, these really, really long DNA molecules, to fit into really small spaces inside the cell. We also have non-coding DNA. Non-coding DNA refers to regions of DNA that do not code for proteins or RNAs, right? That's kind of weird. In prokaryotes like bacteria, only about 12% of the genome is non-coding. But in eukaryotes such as humans, up to 98% of our genome can be non-coding. That's pretty crazy, right? This information does not code for proteins or RNA. These regions are involved, we recently discovered, in regulation, so regulation of gene expression, in DNA packaging, and in stability. And we used to call it junk DNA, but now we know that that's not true. This information or this term is outdated because these areas, these non-coding DNA regions, actually have important regulatory functions. So they somehow regulate gene expression, and we'll learn more about that later. So there is still a lot to discover about non-coding DNA, but so far we know that they are found in both prokaryotic and eukaryotic genomes, and they are regulatory regions that control when and how genes are expressed. Some regions produce non-coding RNAs with structural or regulatory functions, and in eukaryotes we'll learn later on that non-coding DNA can also be made up of structures called introns or spacers, or repetitive elements that are going to influence the structure and stability of our chromosomes. And we also have extra chromosomal DNA. Remember, in eukaryotes, like the one shown here, mitochondria and chloroplasts have their own circular DNA, and this supported the endosymbiotic theory. Some viruses can also maintain their genomes in our host cells. So over here, there is a picture of normal epithelial cells from a pap smear, and then abnormal cells from human papillomavirus that causes certain structural changes. For prokaryotes often carry plasmids that are called extracircular DNA molecules, smaller than their regular genome, which are going to often encode virulence factors that cause disease in humans, or even antibiotic resistance. So these plasmids usually provide traits that are not essential for survival, right? The ones that are essential for survival are in the main chromosome. For plasmids, they can encode additional traits like antibiotic resistance or virulence factors, and these plasmids can be transferred between cells through something known as horizontal gene transfer. So horizontal gene transfer is between cells. Gene or genetic information is transferred between cells within the same generation. This was different from vertical gene transfer, which was the transfer of genes from one generation to the next, like mother to daughter. But horizontal gene transfer can really rapidly spread traits between bacteria, such as antibiotic resistance. Plasmids are also widely used in biotech. We use them as vectors to move genes between cells. And for example, Bacillus anthracis, I'll show you in the next slide, its virulence depends on two different plasmids. So these two species of Bacillus, Bacillus cereus and Bacillus anthracis, are very similar, not identical, but both potentially pathogenic. Bacillus anthracis, which causes anthrax, is much more harmful because it has these two additional plasmids that both encode toxins. This checkpoint asks, how are plasmids involved in antibiotic resistance? So remember, plasmids are those small circular DNA molecules that are separate from the main chromosome, and these plasmids often carry genes for antibiotic resistance. We'll learn later that these are actually called R plasmids, the letter R. And they can transfer between bacteria. These plasmids can transfer between bacteria through horizontal gene transfer, specifically something we'll learn more about later called conjugation, and that can spread antibiotic resistance. So this really provides a rapid way for bacteria to adapt to antibiotic stress. And if we switch gears and look at viral genomes, we know that they vary widely in type and size. We saw that their genomes can be DNA or RNA, single or double-stranded, and their genomes are typically very small since they rely heavily on host cell functions. So they don't have to make that many genes themselves, right? They don't have to have that many enzymes. Genome sizes can range from about 3,500 to 2.5 million bases, and this diversity really enables viruses to adapt to many different hosts and many different replication strategies. We just covered this checkpoint, but viruses can have DNA or RNA genomes, and these could be single or double-stranded. Genome sizes vary from a few thousand bases to a few million, and RNA viruses can actually mutate more quickly than DNA viruses because RNA replication does not have proofreading, which we'll learn later on that DNA replication does. This diversity in viral genomes really reflects the virus's adaptation to different hosts and those different replication strategies. And if we look at the variation in genome size of different organisms or viruses, we can see there is huge diversity. So mammals have fairly large genomes like humans, but they're not the biggest. Some plants or other animals have larger genomes than we do. We see yeast here, a type of fungus, bacteria, here's E. coli. Viruses can be smaller because they have such a small size in the range of nanometers, but some of the virus or viral genomes can be big, such as this large virus, which has a genome bigger than some bacteria. A lot of the genome sizes depend on their metabolic and enzymatic capabilities. So if a bacterium, for instance, is an obligate parasite, it needs to be inside a host cell, then they're going to have small genomes because the host is going to supply most of their nutrients. Whereas if a bacterium such as Pseudomonas aeruginosa has a huge variety of metabolic and enzymatic capabilities, then they're going to have larger genomes. All right, let's recap 10.4. For the first one, a gene is a segment of DNA that encodes a functional product, which can be protein or RNA. A genotype is the complete set of an organism's genes, right? Its genetic makeup. Phenotype is the observable traits or characteristics that are expressed from that genotype. So the genotype really provides the blueprint, but the phenotype is the outcome under specific conditions. For the second one, chromosome structure and packaging. So chromosomes are long, continuous DNA molecules containing many, many genes. In prokaryotes, it's usually a single circular chromosome located in the nucleoid, whereas in eukaryotes, it's usually multiple linear chromosomes located in the nucleus. And DNA is compacted by wrapping around histone proteins in eukaryotes or similar proteins in prokaryotes. And then further coiling and folding really produces a really higher order structure of chromatin so that it can fit into that tiny cell. For the next one, I kind of mentioned it, comparing prokaryotic and eukaryotic chromosomes. Prokaryotes usually have one circular chromosome located in the nucleoid. They don't have a membrane-bound nucleus. And prokaryotes sometimes also have plasmids. For eukaryotes, they have multiple linear chromosomes within that nucleus, and they're organized with those histone proteins to form chromatin. And eukaryotes usually have larger genomes and more regulatory DNA. The last one, extrachromosomal DNA is important in prokaryotes. Remember, those plasmids can carry genes for traits like antibiotic resistance or allow them to produce toxins or other metabolic functions. In eukaryotes, we have extrachromosomal DNA in our mitochondria and chloroplasts, and remember, those have their own DNA, and this DNA can be replicated and expressed independently of chromosomal DNA. This really provides genetic diversity and adaptive traits beyond the main chromosome. That takes us to the end of Chapter 10. In Chapter 11, we're going to look at DNA replication and gene expression. Thanks for watching.